19th. Really glad to see so many of you here, and thank you so much for tuning in from all over. I'm Julia Haggerty. I'm the interim director here at Montana State, and really excited about today's rough cut. Just so you know, this is being recorded, and any technical problems you can direct towards me in the chat. Before I introduce today's speaker, I'll just remind you that we've had a series of talks, of which today's talk is one installation looking at issues in climate science and climate change mitigation and adaptation over the course of this fall semester. And these have been a really wonderful series of talks. This will be our last sort of formal unit in that set. I want to point out to those of you who are teaching or otherwise looking for resources that all of these talks are recorded and archived in a video library on our website. So it's made a really nice kind of collection of pieces that you can share in your classes or refer back to. And on the note of scheduling, this will be our last formal research talk of the fall. We had a cancellation in December, and we've got Thanksgiving next week and then finals after that. So we may try to use those Wednesdays for some internal informal sessions, so stay tuned for that via email. And we are working and already very busy with the spring rough cut schedule. Paige is putting that together, and we should have that posted for you in the beginning of December so you can plan your spring events. So without further ado, I'd like to introduce Dr. Danielle Ulrich, who's an assistant professor in the ecology department. She joined us in 2019 and then promptly got locked away in Zoom world after the pandemic started. So many of us haven't had a chance to meet her in person, but we're so thrilled that she's here. And I'm going to forego the formal academic introduction, given that you've read that in her bio. But I'm going to welcome Danielle, and we're really excited to hear your talk today. So thank you so much for being here.
Great. Thanks, Julia. Thanks, Paige. Thanks to the Rough Cut Seminar Series organizers for inviting me and hosting me today. So I'll get started, and I'm happy to share my research titled A Seedling Eco-Physiological Comparison of 3 High Elevation Pine Species. So let's jump right in. So we know that plants, forests, vegetation are incredibly valuable because of the ecosystem services they provide us with. They provide our food, our clean air and water, healthy ecosystems, and lots of fun, beautiful places to spend time in. And so the value of forests is clearly immeasurable. And we in Montana know this because of the beautiful state we live in. This photo is a photo I took, or this middle photo here is from Glacier National Park from this summer. And as a result of these valuable ecosystem services that forests provide, and in light of our rapidly changing climate, and as a plant physiologist, the goal of my research is to understand, mitigate, and predict the impacts of environmental stress, both abiotic and biotic, on plant function, on plant physiology, and how that underlies survival and mortality. So to do this, I examine physiological processes and responses to stress at the leaf level, whole plant level, and landscape level. My lab's research spans 3 main project areas: seedling ecophysiology of high elevation pines, plant microbe interactions, and adult tree ecophysiology and forest hydrology. This research would not be possible without the help and hard work of undergraduate and graduate students in my lab, including those students I did not have their photos of. So thank you all very much. So today I'm going to share some preliminary results we have from our seedling ecophysiology research on high elevation pines. So what are high elevation pines? Well, for our work, we focus on whitebark pine, or Pinus albicollis, or what I'll refer to as pile, limber pine, Pinus flexilis, or piffle, and Great Basin bristlecone pine, or pilo. These are 5-needled pines, meaning they have bundles or fascicles of 5 needles. They inhabit high elevations anywhere from 5,000 feet to an upwards of 11,000 feet in elevation. These pines are slow growing and they're very long lived. Pile can live to about 1,300 years old. Piffle can live to about 3,000 years old. And Pilo can live up to and upwards of 5,000 years old, with Pilo being the oldest non-clonal organism, nearing, I think Methuselah is now nearing 5,000 years old. These species inhabit the western US, as shown from these species distribution maps. You can see that Pile on the left has the largest and most northern distribution, where Pilo on the far right has a smaller distribution restricted to the Great Basin region of the US. Here in Montana, we have both whitebark and limber pine, and in the Greater Yellowstone ecosystem, whitebark is often found at higher elevations than limber pine. So why study these pines? Why study these species? Well, these long-lived, slow-growing pines are very ecologically important, and they are foundation and keystone species. They're foundation species because they're often the only tree species to establish at such high elevations, and therefore the first species to establish at these sites. They therefore form and define these habitats for other organisms. These species are keystone species because they are depended upon by other organisms. For example, their seeds serve as a nutritional food source for the grizzly bear and the Clark's nutcracker, shown here, and other small mammals. In addition, these pines are known to protect snowpack and prolong stream flow into the growing season, supporting downstream aquatic ecosystems, thinking rivers and fish. Unfortunately, these ecologically important species are declining at alarming rates. These pines are threatened by a suite of factors, including white pine blister rust, which is a non-native fungal pathogen, bark beetle infestation, wildfire, competition with more shade-tolerant conifers, and these pines are also threatened by climate change, by warmer, drier conditions. As such, recently this year, whitebark pine has been officially listed as threatened under the U.S. Endangered Species Act as of February of this year. To combat these declining pine populations, outplanting seedlings is the primary restoration and conservation strategy. The seedling, however, is the most vulnerable developmental life stage, given their small stature, which means they have an increased susceptibility to environmental stress and physical damage. It's critical, or because seedling establishment and survival drive species distributions where species are found, it's critical that we understand mechanisms and traits that underlie seedling establishment and survival. And this is because understanding seedling physiology can inform our understanding of current forest boundaries and improve predictions of future forest boundaries. There's much information and research documenting adult mortality of these pines, which is why we know these pines are declining at alarming rates, which contributed to whitebark being listed as threatened under the Endangered Species Act. However, there's relatively little information on seedling establishment, including traits underlying seedling survival, such as stress tolerance, photosynthetic performance, and other growth-related traits. Yet, we need this information to better understand where best to outplant these seedlings for restoration and to better understand foundational seedling regeneration processes. And in general, these pines are relatively understudied compared to economically important species like Douglas fir, lodgepole, ponderosa. So there's a great need to study the seedling physiology of these threatened species. So that brings me to the research questions of this work that we'll examine today. First, what are the seedling morphological and physiological traits of pile, piffle, and pilo? Second, how do seedling traits relate to each other? What are the trait-trait relationships? Are there any trade-offs between traits? And third, how do seedling traits relate to climates of origin? Do seedling traits affect their contrasting geographic distributions? Will one species better establish under warmer, drier conditions? So to answer these questions, we obtained seedlings donated to our research by the Coeur d'Alene Nursery of the Forest Service. The Coeur d'Alene Nursery grows these pines from seed with the goal of outplanting these seedlings on ranger districts throughout the Forest Service Region 1. The nursery was generous enough to donate a subset of these seedlings, diverting them from outplanting and instead donating them to our research. And for that, I'm very grateful. We obtained these seedlings from the nursery. We transplanted bare root seedlings to pots of soil in MSU's Plant Growth Center greenhouses, which is shown here. We received 5-year-old pile, piffle, and pilo seedlings, as well as pile of different ages and some other age classes and other populations. But today we're focusing on the 5-year-old pile, piffle, and pilo. This map shows the geographic distribution of whitebark pine in blue, limber pine in pink, and bristlecone pine in yellow. And the circles indicate the seed source locations of the 5-year-old seedlings that were donated to us from the Coeur d'Alene Nursery. And we received 2 families of each species, indicated by 2 circles per color, two circles per species. These are the climate conditions of the seed source locations and the number of seedlings planted in the Plant Growth Center of each family and species. We have a pile in blue, piffle in pink, and pilo again in yellow. And we can see that we have a range of numbers from 16 up to 53 seedlings that were donated to this work. And in general, we can see that pilo in yellow originated from relatively higher elevations and drier climates than pile and piffle. There's lower precipitation and a higher vapor pressure deficit, or VPD maximum VPD. And in general, pile and piffle were from lower elevations and from wetter, colder climates. Here are photos of the transplanted seedlings in the Plant Growth Center greenhouse. You'll notice the morphological differences in crown architecture or branch arrangement of the 3 species. And this is notable and remarkable because these species were grown from seed in the same conditions, whether that was in the Coeur d'Alene Nursery or here at the Plant Growth Center. Yet we still see these very distinct branch arrangements. And in another project that I won't be presenting today, we are comparing the crown architecture of pile and piffle, which is work led by undergrad student Megan McFadden. So to answer our 3 questions, we measured 32 traits that are categorized into these trait categories that I've listed here. And this is a list of the 32 traits, which I don't expect you to read each type of trait, so I'll summarize each trait category. So first, morphological measurements. Morphological measurements consisted of needle size, stem height, and base stem diameter of the seedlings. Stomatal traits included the size and density of the stomata on the needle. Now, stomata are tiny pores on needles where plants take in CO2 for photosynthesis and lose water through transpiration. This CO2 uptake is important for photosynthesis, while the water evapotranspiration through the stomata is important because this drives water transport through the seedlings. The size and density of stomata then can relate to a plant's regulation of these gases in and out of the plant, and can tell us, for example, about how much photosynthesis a seedling may do and how that might affect growth rates. So for example, larger stomata can let in more CO2 for photosynthesis, which may lead to increased growth. Bud burst phenology here refers to the number of days to reach a particular bud burst stage. So every year, a new needle growth starts as a bud and then eventually leads to needle growth or needle elongation. Fewer days to a bud burst stage indicate earlier bud burst, while more days, a greater number of days to a bud burst stage, indicate more delayed or later bud burst. Bud burst phenology can tell us about when during the season the seedling does most of its growing. For example, delayed bud burst means that needle growth occurs later in the season, while earlier bud burst means that needle growth starts earlier in the season. Photosynthetic capacity, drought, highlight, heat tolerance, and cold tolerance are what I refer to as these physiological traits, where photosynthetic capacity refers to the physiological capacity to do photosynthesis. And this is related to rubisco content and protein and enzymatic activity and capability to do photosynthesis. These tolerances to drought, highlight, heat, and cold refer to stress tolerances of needles. Higher tolerances indicate a greater ability to withstand each of these stressors. In this long list of these 32 traits, I also have this rightmost column of sample size, where sample size for all of these traits range anywhere from 5 to 27 individuals per family. And that's because morphological, stomatal, and bud burst measurements were much easier and faster to make on a greater number of individuals than the physiological measurements, which are much more time consuming and more difficult to make, making the sample size smaller. So most of our physiological traits were conducted on about a sample size of 5. Okay, so back to our questions. What are the seedling morphological and physiological traits of these 3 species? So here's our list of our 32 traits again, with an added column for p-value. And instead of, again, having you read that list, here I'm highlighting in blue the traits and trait categories where we found significant differences among the 3 species using a one-way ANOVA. In general, species differences were observed in morphological, stomatal, bud burst, and cold tolerance related traits. So next we'll look at each of these 4 categories of traits and see how the 3 species compared to each other in each type of trait. So first, morphological measurements. Morphological measurements of stem height, stem diameter, and needle length were measured by undergrads Jack Turney, Jesse Irish, and Rachel Robin. Different letters in each of these plots indicate significant differences between the 3 species using a Tukey's post-hoc test. And in general, we found that Pilo, or Great Basin bristlecone in pink, was taller, had a greater stem diameter, and shorter needles compared to whitebark and limber pine, compared to pile and piffle. We see that piffle or limber pine had the shortest stem height, while pile or whitebark had the smallest stem diameter. Our second category of traits where we saw significant species differences was stomatal traits. So stomatal traits were measured by undergrad students Megan McFadden and Rachel Robbins. And to do this, we took imprints of needles and stomata using clear nail polish, letting it dry, and then using a clear piece of tape to affix that dried nail polish imprint to a microscope slide and viewing the stomata under a microscope. And so what I'm showing here are example images of stomata of the 3 species. And these images are notable because we can qualitatively see differences between the 3 species, which is really cool. And so we measured stomatal area or stomatal size using these images, kind of the area of the stomatal opening, and stomatal density, where we counted the number of stomata per length of needle. So we're looking at 2 rows of stomata in each of these species. And for example, limber pine was notoriously difficult to get stomatal imprints from, whereas Great Basin bristlecone had these very nicely defined stomata that made it really easy to quantify. So what did we find in terms of stomatal traits? We found that Pilo bristlecone had greater stomatal size, larger stomata, greater stomatal area, and greater stomatal density, whereas Pile had smaller stomata and lower stomatal density, fewer smaller stomata in whitebark. Piffle had larger stomata than whitebark and the same stomatal density as Great Basin bristlecone pine or Pilo. So as we're seeing in Pilo, these more larger stomata in Pilo may indicate more CO2 uptake for photosynthesis and possibly more growth, which is what we saw in that greater height of pilo in the previous morphological traits. The third category of traits where we saw significant species differences was budburst phenology. Budburst phenology measurements were led by undergrad student Franklin Alonji. We identified 6 stages of budburst, indicated here, stage 1, 2, 3, 4, 5, 6, from a tight bud all the way to needle elongation. Now, budburst timing can tell us when seedling needles do most of their growing. Earlier budburst indicates growth earlier in the season, while later, more delayed budburst indicates growth later in the season. We found that pilo, in pink again, had the most delayed budburst, while in contrast, piffle limber pine had the earliest budburst. And we assessed this as the number of days to reach budburst stage 5. So it took pilo more days to reach this budburst stage. It took piffle fewer days to reach that budburst stage. And pile had an intermediate number of days to reach that stage. This suggests that pilo starts its growth later in the season, while piffle starts its growth earlier in the season. And this is based on the timing of when that needle elongation starts. The fourth category of traits where we saw significant species differences was cold tolerance. Now, cold tolerance measurements were led by master's student Sean Hoy-Skubik. Sean measured cold tolerance in terms of cold temperature effects on both photosynthetic performance, which is in this top panel, and cold temperature effects on cellular damage, which is in this bottom panel here. In both independent methods of cold tolerance measurement on photosynthetic performance and cellular damage, pilo was least cold tolerant. Pile and piffle were significantly more cold tolerant than pilo. And this kind of makes sense because pile and piffle, as we saw, originated from colder climates than pilo. And finally, we measured tolerance to heat, high light, and drought, as well as photosynthetic capacity. And these physiological traits, as I referred to them, were led by master student Chloe Wastanes. These traits were measured with the LICOR, the portable photosynthesis instrument that is equipped with infrared gas analyzers. And the LICOR was used to measure heat, high light, and photosynthetic capacity, or heat and high light tolerance and photosynthetic capacity, while the pressure bomb shown here was used to assess drought tolerance. I'm happy to discuss in more detail the methods we used to assess these if anyone has questions at the end. And we found no significant differences in these physiological traits among these species. I do show them though, because notably, high-low Great Basin bristlecone pine has slightly lower, even though it's not significant, slightly lower heat tolerance compared to pile and piffle. And if you recall, pilo also had the lowest cold tolerance, or lower cold tolerance than pile and piffle. Okay, so we looked at how the 3 species kind of differ in their morphological and physiological traits. Next, how do these seedling traits relate to each other? What are some trait-trait relationships or trade-offs among traits? So we identified trait-trait relationships using this correlation matrix shown here. This correlation matrix contains all 32 traits, which I don't expect you to read. I'll kind of walk you through this, and we'll highlight some relationships of interest. The circle size in the correlation matrix represents Pearson correlation coefficients, where the larger circle indicates a larger correlation coefficient. Circle color indicates the direction of the correlation, where red is a negative correlation and blue is a positive correlation. If circles are present in the boxes, that means that the correlation was significant at a p-value of less than 0.05. If there's a blank in the correlation matrix, it means that the correlation was not significant and is not shown in the matrix. For today, we're going to focus on the morphological, stomatal, bud burst, and cold tolerance traits. Those traits we found to have significant species differences. I've highlighted some of those trait-trait relationships in black boxes shown here. And so next we'll visualize these trait relationships outlined in these boxes. Specifically, we'll focus on trait relationships between stomatal area, height, bud burst, and cold tolerance. Here are those trait relationships that were outlined in those black boxes. The small circles in all of these panels are individual seedling replicates for which both measurements on the Y and X axes were made. The larger circles with standard error bars represent species means. The line represents the linear regression or correlation of the individuals of the small circles. And the gray shading represents the 95% confidence interval. In the top two panels, we see that bud burst delay or number of days to stage 5 and stomatal area both increase with increasing stem height. In the bottom plot, we see that cold tolerance in terms of photosynthetic performance on the y-axis here decreases with increasing stomatal area. And as we saw in the box plots in question 1, we see a separation between Pilo or Great Basin bristlecone pine and the other two species, Pile and Piffle. We see that Pile is taller, has larger stomatal area, has a more delayed bud burst, and also a lower cold tolerance. A slight tangent here, but of note, we found a non-significant relationship between cold tolerance on the y-axis and heat tolerance on the x-axis shown here. Despite the lack of this significant correlation among individuals, we can see how cold and heat tolerant species means are greater in Pile and Piffle compared to Pilo. Again, these are species means with standard error bars in these larger circles. So we can see how Pilo kind of separates from the other two species, where Pilo is less cold tolerant and less heat tolerant. Okay, so we just looked at trait-trait relationships. What about trait-climate relationships? How do seedling traits relate to climates of origin? Do seedling traits affect the species' contrasting geographic distributions? Will one species fare better under future warmer, drier conditions? So we recall that Pilo, or at least the seedlings we obtained from the nursery, originated from higher elevations, warmer temperatures, and drier conditions. We have also, up to now, have observed that Pilo is taller, has larger stomata, exhibits delayed bud burst, and lower cold tolerance. And this pattern I want to highlight because it drives our climate trait relationships shown in this correlation matrix. Again, the numbers, rather than circles this time, the numbers are Pearson correlation coefficients. The number, or the color of the number, again, indicates the direction of the correlation. And if the numbers are present, the correlation was significant. If they're absent, if there's a blank, that means that the correlation was not significant. And generally what we see is that seedlings that are taller, have larger stomata, delayed bud burst, and have lower cold tolerance are associated with higher elevations, warmer temperatures, and drier conditions. And for the purposes of today and to kind of simplify, we will next visualize relationships between our 4 traits, height, stomatal area, bud burst, and cold tolerance, with, for simplicity and time, just 3 climate variables that I've outlined here in these boxes: elevation, precipitation, and minimum temperature. And I've shown only, and up till now also, I've only been showing 5 climate variables for simplicity, but we know that we can get many, many more climate variables than this. But for today, I would like to show examples of some climate trait relationships in just these 3 climate variables, in just elevation, precipitation, and minimum temperature. Second. Sorry about that. Okay. Okay, so we're going to focus on our climate trait relationships for elevation, precipitation, and minimum temperature. So first, elevation. I have height, stomatal area, bud burst delay or number of days to that stage 5, and cold tolerance in terms of photosynthetic performance on the y-axis, and our climate variable of interest, elevation, on the x-axis. And we can see that height, stomatal area, and bud burst all increase with increasing elevation. While we see that cold tolerance slightly decreases with elevation, although we see that it's only marginally significant with a p-value of 0.056. That second climate variable I said we're going to look at is precipitation, where now I have precipitation, total annual precipitation in millimeters on the x-axis with those same 4 traits on the y-axis. And we can see that height, stomatal area, and bud burst delay all decline with increasing precipitation. We see that cold tolerance increases with increasing precipitation. Or in other words, under drier conditions, we see taller height, larger stomatal area, more bud burst delay or greater delay in bud burst, and lower cold tolerance. The third and final climate variable I said we're going to look at is minimum temperature, or Tmin, on the x-axis here. And I note it even though it's marginally significant at a p-value of 0.06, but I note it because we see a slight decrease in cold tolerance with increasing minimum temperature, or cold tolerance increases in families from colder climates, which is what we might expect. Okay, so it's clear that there are many traits that we can measure, that we've measured in my lab. I've shown you 32 today, but there are many more as well. And we also know that there are many climate variables we can look at besides just the 5 that I've presented today. And also plenty of places we can get climate variables from, including PRISM, World Climate, Climate NA. And so all of these traits, all of these variables are unending, which kind of makes identifying and understanding trait-climate relationships daunting and challenging. So to address this challenge, I wanted to find kind of a single trait, if there was one, a single trait to relate to climate variables, which might, for example, help explain the contrasting species distributions and help us better understand trait-climate relationships. If we could kind of condense all of these traits maybe down to one. And so to do that, I was inspired by this study that was published in 2016 by Scheel, Banzel, and co-authors in Ecology and Evolution, where they used principal components analysis to reduce multiple physiological traits down to one metric or one PCA dimension. And they related that single metric, that PCA dimension, to climate variables to help predict future species distributions. And so I tried a similar approach with our data. And now this is PCA is common in other environmental fields, but it is novel and not normally done in the field of plant physiology. But it seemed like a great strategy for condensing all of our variables or traits down to one metric and examining its relationship with climate. And it's something new I wanted to try and possibly get feedback on today. So PCA, Principal Components Analysis, is a dimensionality reduction method that transforms a large set of variables or traits into a smaller one that still contains most of the information in the larger data set. So as a result, dimensionality reduction trades a little accuracy for simplicity, which is kind of what I was looking for. Or in other words, this reduces the number of variables of the data set while preserving as much information as possible. And again, I focused on those same traits where we found significant species differences. I focused on the morphological, stomatal, bud burst, and cold tolerance traits. And in PCA, I learned that all individuals that you're analyzing must have the same traits or measurements made on them. And there can be no blanks or no missing data, which was a bit challenging with our data because not all traits, not all the physiological, morphological, not all of the traits were measured on the same exact individuals. And so given that limitation and given the focus on these 4 categories of traits, I was able to whittle down our data set to 21 individuals, 21 seedlings across 3 species that had the same morphological, stomatal, bud burst, and cold tolerance measurements made on them, and conducted PCA on that. Here is a PCA scree plot that shows the number of dimensions on the x-axis and the percent of variation or variance explained by each dimension on the y-axis. And note that dimension 1 explains about 35% of the variance, while dimension 2 explains about 20% of the variance. So here's a PCA biplot of dimensions 1 and 2. Remember, dimension 1 explained about 35% of the variance. Dimension 2 explained about 20% of the variance. Traits are in black here. The arrows are vectors whose length indicates how strongly each trait influences a principal component, influences a dimension. The angles between the vectors that are less than 90 degrees represent a positive correlation, and angles between the vectors that are greater than 90 degrees represent a negative correlation. So for example, if we looked at needle length and height, which is an angle greater than 90 degrees, you can see that needle length and height are negatively correlated, which makes sense because we did see shorter needles and taller height in bristlecone and pilo, for example. So we can nicely see with this plot how these three species separate along these 2 dimensions. And as we've seen in the box plots in question 1 and in the correlations in question 2, we can see that pile and piffle in blue and yellow here kind of cluster together more closely, while pilo, or Great Basin bristlecone pine, in pink is more different than the other 2 species. The closer that points are together, the more similar they are, whereas the further apart the points are, the more different they are. And we're seeing that a species kind of separation nicely in this space. The smaller points represent the individual seedlings, those 21 total, and the larger circles represent the centroid of each of the species ellipses. Next, I identified the top 4 traits that were most responsible for the species separation, or the top 4 traits that had the highest contribution to the separation between the 3 species. And these were stomatal area, height, bud burst, and cold tolerance. And again, though, I was seeking to condense these down to one metric to relate to climate. And to do that, I focused on dimension 1, highlighted in yellow here, as the metric to represent our suite of traits and to plot against climate variables to examine trait-climate relationships. Here in this correlation matrix, we see that we've got our 4 traits: height, stomatal area, bud burst, and cold tolerance, and our 5 climate variables: elevation, precip, minimum temperature, and maximum vapor pressure deficit. And we can see generally with the presence of these numbers that there were significant correlations between these traits and these climate variables. But again, I was seeking to condense our traits down to one dimension and focus on dimension 1, highlighted in yellow here. So I wanted to use dimension 1 to represent our various traits. And so next we'll visualize the relationships between dimension 1 and these 4 climate variables with significant correlations with dimension 1. So that's elevation, precipitation, maximum temperature, and maximum BPD. And so here we are visualizing the significant correlations between climate variables, those 4 climate variables, and dimension 1. Have elevation, precipitation, maximum temperature, and BPD max, or maximum BPD, against dimension 1 on the y-axis. And we have some significant correlations, which is cool. And we're able to see how Pilo in pink separates from Pile and Piffle using this dimension 1 to represent our various traits in this single metric. Again, and we see that Pilo is found at higher, warmer, drier climates than Pile and Piffle, and which we can see in these different plots where Pilo kind of groups here on this end of elevation in dimension 1 and precipitation, indicating Pilo is at higher elevation from drier climates and from warmer climates. And so we're able to look at these relationships with this dimension 1. And so this was a little test experiment to see if we could reduce our traits down to one metric and relate to climate. And so perhaps in the future, such a metric could be used to predict physiological traits and or responses to future climates. And it's something I hope to explore more. Okay, that's a wrap. So what did we learn? We answered these 3 questions and started with the first question of what are the seedling physiological, morphological traits among our 3 species, Pile, Piffle, and Pilo. What did we find? We found minimal species differences in physiological traits, which was unexpected. So in minimal differences in photosynthetic capacity, drought, heat, and high light tolerance. While in contrast, we actually found stronger species differences in morphology, bud burst, and cold tolerance traits. We observed Pilo to exhibit delayed bud burst. Pilo was also taller and had larger stomata. And one interpretation is that Pilo relies on monsoonal rain during the growing season for its fast growth. And this characteristic is notable because it appears to be species specific or genetically determined, because we've observed these patterns in these seedlings that have grown from seed in the same environments. So we see kind of these perhaps a reliance on monsoonal rain reflected in traits even when grown in common environments. And so the delayed bud burst indicates that most of that needle growth is happening later in the season, which may coincide with precipitation that falls in the form of monsoonal rain later in the summer. We also saw that Pilo was taller and had larger stomata, which could indicate greater CO2 uptake, faster growth rate, which we saw with that taller growth, and that it might grow faster when water is available during those warmer months. And we know that warmer temperatures can promote or increase growth. So if water is available, warm temperatures available, big stomata taking a lot of CO2, then maybe a lot of growth is happening. And that's what leads to that much taller height, that delayed bud burst, larger stomata that we observed in Pilo. On the other hand, Pile and Piffle were more cold tolerant. They were shorter, much shorter in stature, and they exhibited earlier bud bursts, suggesting that they start their growth earlier in the growing season, which may make sense because rather than monsoonal rains like Pile and Piffle may rely more heavily on precipitation from spring snowmelt. And so they may grow slower during the spring because it's colder, so they are more cold tolerant and are shorter statured. So this might explain that early bud burst in Pile and Piffle that we observed to take advantage of or start growing with that spring snowmelt. And again, the shorter stature and possibly slower growth of these species may be due to the colder temperatures that these species experience compared to Pilo. We saw that Pile and Piffle did originate from climates that were colder than Pilo. And we know that cold temperatures can limit growth which may contribute to them being shorter or growing slower. We answered the second question about trait-trait relationships and how seedling traits relate to each other. And we saw that taller seedlings exhibit lower cold tolerance, have larger stomata, and delayed bud burst. And this was largely due to the species differences, particularly driven by phyllo. And finally, we looked at trait-climate relationships, looking at trait-climate relationships with our measured traits, and then trait-climate relationships using our dimension 1 from that PCA analysis. And overall, we found that we knew phyllo originated from drier, warmer climates of origin, may rely on monsoonal rain, and were taller, larger, had larger stomata, delayed bud burst, and lower cold tolerance. Phyllo and piffle, on the other hand, originated from wetter, colder climates and may rely more heavily on snowmelt, on spring snowmelt for water, and were shorter in stature. They had smaller stomata, exhibited earlier bud burst, and higher cold tolerance. And so how will climate change affect these species? Well, since it appears that perhaps the patterns we're observing indicate that these species may rely on different types and timing of rain, of precipitation, either monsoonal rain or snowmelt, the effect of climate change on each of these species will depend on how much climate change affects monsoonal rain and snowmelt. For example, if we see greater changes in monsoon rains, then maybe we'd predict a greater effect on phyllo. Whereas if we see a greater effect on snowmelt, then we might see a greater effect of climate change on phyllo and piffle. So future work, where are we headed? We're headed in lots of directions. We are continuing our work looking at climate physiology relationships between the three species, between phyllo age classes that we have, and between phyllo populations that we have. And that work is led by a master student, Chloe Wastenese. We are also looking at whole plant responses to climate change type stressors, such as heat and drought stress, in phyllo, piffle, phyllo, and also in PIBA, which is foxtail pine. And this work is led by Sean Hoy-Skubik, Jess Harris, and Katie Sparks, master's students. As I noted earlier, we noticed the striking differences in crown morphology between the seedlings, and we are examining that more closely, differences in crown morphology between phyllo and piffle. And this work is led by undergrad student Megan McFadden, who is a visiting summer intern from Whitman College. We are also, as we continue to torture and destructively harvest and sample these seedlings, we are reducing the number of seedlings we have these days. And so we are actively planting more seed to try to replenish that. And we're planting seeds of phyllo, piffle, phyllo, subalpine fir. And these are seeds from different climate zones and also a range of rust resistance and rust susceptibility. And that work is led by master student Jess Harris. And this is a picture that Jess took yesterday of an emerging limber pine seedling, which we're very excited about. And we're also looking at the effects of adult whitebark mortality on snowpack and stream flow and on forest hydrology. And that work is led by Tio Rautu, master student, who's using water balance modeling and field-based approaches to answer this question. And with that, I thank you all for your attention and time. And I'd like to thank the Coeur d'Alene Nursery for donating the seedlings to our research, MSU's Plant Growth Center and David Bombauer for their unending support. I'd like to thank various funding sources, the Forest Service Forest Health Protection Program, the Whitebark Pine Ecosystem Foundation student grants, SICA Ecosystem Grant, a grad student grant, Montana Native Plant Society, MSU's Undergrad Scholars Program, and the Whitman College Internship Program, as well as all of my excellent Ulrich Lab members. Without them, this work could not have been done. So I thank you all very much. And with that, I'd be happy to take any questions.
Wonderful. Thanks so much. I'm going to encourage our audience. It's just such a rich and full talk. Thank you, Dr. Ulrich. You can Use the emoji reactions button to raise your hand and to give a round of applause. And you're also welcome to enter questions in the chat. I know that there was lots of really interesting work presented here, so look forward to, we've got about 10 minutes for questions and answers.
Great, thanks. I'm trying to pull up I'm struggling finding this chat.
Don't worry, I
stop
can
sharing
see
my
it.
screen?
Keep track of it. I don't know. I think it will depend on if people want to go back to some of your data slides.
Oh, here's chat. Okay.
Rush to it right now. Would anyone like to start us off with a question? There's one that came up in the chat from Yuriko Yano, who asks, is there a temperature threshold for bud burst? If so, would the thresholds differ across the 3 species?
That's a great question. I do not know if there is a temperature threshold for bud burst. In our case, I think possibly not, only because all of these seedlings were grown in the same environment in the PGC greenhouse, and yet we still did see these species-specific differences in bud burst timing, where Pilo just really took its time. If so, would the thresholds differ across the 3 species? And I mean, I guess it could be temperature related, but since we saw it in this common environment, it kind of suggests that it could be more genetically determined rather than environmental temperature determined. But it could be indirectly related to temperature based on the environment experienced by parent trees or something, for example.
Go ahead, Kathy.
Yeah, Danielle, it's so cool to see this, and I love this experimental work to try to understand the climate sensitivities of these species. And just as you were talking, I'm thinking about the paleo record of these species and what I know about it. And the interpretation that you offer for bristlecone seems very counterintuitive to me, because we know that during the last ice age, it was much more widespread in the American Southwest, you know, growing down to low elevations, filling the desert areas. And so that was a time when there was no late summer precipitation and there was very low CO2, you know, 2/3 of present day. And I suspect the cold tolerance relationships that you're finding in your experiments wouldn't well explain what we see in the paleo record. So I don't know what you think about that, but it's like, it would be really cool to try to kind of jive these 2 sources of information, your experimental stuff with a sort of long-term perspective.
Yeah, that's super cool. Thanks, Kathy. Yeah, that's a great point. You know, maybe something that I haven't focused on here is, you know, how we focus on these 5-year-old seedlings, but how do those physiological stress resistances, those tolerances change with age? So maybe that's just what I'm seeing at the seedling stage, and perhaps may not be as important as I think or hope it is for what we see at the adult stage. So yeah, that's I like also the comment about lower CO2 at that time as well. And so it kind of makes me think, was there something specific about the seed source locations of the Pilo that we obtained that happened to be particularly favorable, or yeah, or I guess, I mean, over the paleo record too, that's many, many years. So things maybe.
Well, during the last glacial period, when the distribution of bristlecone was so widespread, it was cooler, probably, you know, 4 to 5 degrees centigrade cooler. And it was also wetter, but it was probably winter precipitation because of the way the jet stream was reconfigured during that time. So I don't know what the answer is, but it's just kind of interesting to me.
Yeah, I'll be pondering that some more as well. So yeah, thanks for bringing that up.
Diane has a question. Dr. Dubinsky, excuse.
Thanks. Hey, great talk, Dr. Ulrich. I really liked how you separated all those variables and then used the PCA to make a combination of some of these different variables. It's an interesting approach. One question I had was, you talked a little bit about the crown morphology, and can you tell us a little bit more about how you might speculate that crown morphology would affect each of these species' abilities to respond to some of these extreme conditions in the future? Thanks.
Yeah, so in other systems, all else equal, the arrangement of branching can alter how a plant absorbs light. And plants need light for photosynthesis, and so it's been shown in different systems that that plants will spread out more of their leaves or their crown architecture to absorb more light, and so you'll kind of see that in more shade-adapted species. Whereas species that are more highlight adapted that can afford to overlap and self-shade itself is more adapted to highlight conditions because it's not trying to maximize light capture. So in the case of pile and piffle, we kind of see a more spread out arrangement in pile and whitebark, whereas we see this very clumped, overlapping, self-shading, architecture of limber pine. Like when we got them, they just looked like these little bushes, these little like balls of needles. And so it, on its own, and all else being equal, that kind of suggests that perhaps limber pine is more highlight adapted than whitebark is, because it can afford to self-shade itself, whereas plants would not do that. They would do everything they can to maximize light capture. So that's the kind of general direction there. I don't know if you had a more specific question than that.
And can I just ask, how much flexibility is there among individuals in that kind of, I won't call it behavior, but what would you call that? Just the configuration of the way the needles are spread out or not? How much variation among individuals in a species is there such that there could be some potential adaptation?
Yeah, I mean, we can see it within an individual tree, an individual plant. A shade branch a shade conifer branch has more flat needles, where a sun-adapted branch will have more needles with steeper angles to reduce that light capture. Whereas those shade branches have the flat needles to maximize light capture. So within an individual, there's quite a bit of variation, and I was surprised to see such strong differences in crown architecture in these seedlings that were grown in common environments, because I would think it's something that's very environmentally determined, but we didn't really see that here, where there are just these very strong differences. And in fact, with limber pine, we had those two families of limber pine where Franklin, one of the students in my lab, joked that he could tell the difference based on the crown architecture between the one limber pine family and the other limber pine family, just because one was slightly more clumped than the other. So before this work, I would have said that it's very environmentally determined and would be plastic in that way. I mean, once the crown architecture is created, it wouldn't change, but would be very environmentally determined. But we're seeing it seems to be very species specific, or maybe more genetically determined than I had originally thought.
Thanks so much.
So, Dr. Ulrich, there are two questions left for you in the chat in the couple of minutes we have left. I'm going to read them both, and you can decide which order you want to take them. The first is from Dave McWethy, who asks, why do you think pile has a smaller stomata in the generally higher, cooler environments? One might expect larger stomata to take advantage of a short warm season. I'm going to stop with that one. The next one has to do with distribution.
Yeah, thanks, Dave. Also the stomatal measurements were also kind of not what I expected either. But thinking about the pilo, if pile is growing slower, maybe this having smaller stomata is okay, because they are already perhaps grow slower, maybe do less photosynthesis, because they're in these colder climates. So there's not quite as much of a demand to grow so much, because cold is kind of limiting how much growth can happen or how fast growth can happen. In addition, having smaller stomata can reduce water loss, which can be really important for in drought adapted climates. So that's an advantage of having smaller stomata is just like a drought response. But I say that, but it still a little perplexes me because I would expect it maybe to see the opposite pattern in smaller stomata and pi low that's found in drier climates. So we're still kind of digging into it. So it's a great observation and question that we're still thinking about. So thanks.
I'm going to squeeze in one last question, which comes from Anna Shuttle, who asks that knowing that you didn't see large differences between PIFL, sorry, the acronym's not coming to mind, and pile physiology, but the two species tend to grow in different habitats in parts of their range. So what are your thoughts on factors that contribute to their distributional differences despite some physiological similarities, I think is the question there.
Yeah, it's a great question. And this is what I wrap my brain around a lot and think about a lot. I was very surprised, like you point out, not to see any strong physiological differences between pile and piffle. But it seems like something like morphology, like that crown architecture, might be more or response to high light conditions, might be more driving maybe differences in distributions, because that's kind of the big difference we're seeing. And I mean, then other factors that don't relate to the individual physiology could be at play too, that we're not accounting for. So distribution or dispersal mechanisms that might differ. But yeah, I don't know. I'm not positive, and definitely still thinking about it because it's not what we had expected. But the thing we do see the differences in is the morphology and maybe the stomatal traits too. So less physiology, more morphology, which is surprising. But yeah, Anna, we could chat more about it at some point.
Well, this has been wonderful. You can see, like all good research, this is opening up lots of opportunities to talk more with your colleagues, especially on over disciplinary kind of lines. So thank you so much, Dr. Ulrich. It was a wonderful talk. We'll let everybody sign off. If you'd like to take yourself off mute and give Dr. Ulrich a round of applause, that would be great as you exit. So thank you so much.
Thanks, Julia.